Hi everyone, welcome to Basic Science series. Today we will discuss how to purify proteins from bacterial cells. So the topic is bacterial protein purification. We can also rename this topic as recombinant protein purification, but simply you can call it as a bacterial protein purification when you're using especially the E. coli cells. So in this, the main word is the proteins. What are proteins? Proteins are basically biomolecules. They're made up of amino acids. Amino acids are the building blocks of the protein, and there are n number of proteins. Especially if we talk about human cells, human body. We are made up of proteins. We are made up of lot of different kinds of proteins, and they have biological functions. And when they perform these functions, our cell survives and we live. So basically, proteins, they play an essential role in the biological functioning of the cell. And to study the function, we need to purify these proteins. And to purify the proteins, we need host cells. There are different types of host cells. We can use bacteria, we can use yeast, we can use mammalian host, especially the human cell lines, cancer cell lines, we can use insect cell lines and plants, etc. In this particular video, I'll focus on bacterial as a host system to purify protein. We'll discuss in detail about the major steps that are involved in the protein purification from bacteria. We'll discuss the basics also. That what is the principle of the protein purification in bacteria. So first thing is we need a plasmid. Plasmid is a double standard DNA molecule, circular DNA molecule that has the gene of interest. So here I'm writing YGOI, which is your gene of interest in it. In addition to that, there are various components. But another important component is the selectable marker, using which you can screen out the cells where you have the plasmids. First step is to transform the plasmid into the bacterial cell. That is called the transformation. Whenever the extracellular DNA that is getting entered into the cell is called the transformation. Into the bacterial cell is the transformation. When you are doing the same thing in eukaryotic cells, mammalian cells, it's called transfection. So the process in case of bacteria is heat shock method. So you can basically first cool down the cell in ice bath and then expose the cells with the DNA at 42 degrees centigrade for 45 minutes. This time is important here. It will increase or decrease the transformation efficiency. So you have to be very, very careful when doing this experiment. You need to be precise, especially for the heat shock step. Next is, once you do the heat shock, you have to grow the bacteria. After that, you will plate, or you will put the bacteria onto the media plate where you have a specific molecule, in this case antibiotic, because the selectable marker in this case is the antibiotic resistant gene. So if the plasmid is there in the bacterial cell, the bacteria will grow. So you have to be very, very careful. In this case, I have one plate with the ampicillin, because we have the MPR gene, and where we will see colonies, and another without antibiotic. So what will happen in that case, that all the cells are there going to grow with or without plasmid. So you will have lot of different cells in there. It will be too crowded. That is why we need selectable markers, so that we can quickly screen out those cells where we basically have the recombinant plasmid. So we want to remove the possibility of unwanted cells that are without plasmid to select or to process further. Next is, when you do the cloning, you need to first, in this case we are doing the expression, so blue-white screening will not work in this case, but you need to see that video where I have explained the blue-white screening, and that will again strengthen up your concept. Then once you have the cell with the plasmid, you will do the batch growth. You can do up to 1000 liters, up to you. But in the lab, I would recommend you to basically start with 100 ml, 10 ml, or even 1 ml. It depends upon you if you are just checking the expression. But if you are purifying the protein, at least 1000 ml or 500 ml should be good enough. And then plasmid will produce the protein in presence of IPTG molecule, because IPD induction is immediately going to work, and then cell will be hijacked to produce the recombinant protein. After that, protein will be expressed. So if you have the his tag with the gene of interest that you're looking for, then you can simply use nickel column chromatography to purify the protein. After that, what will happen, that protein will be purified, and then that purification, the amount of the status of the purification can be checked by SDS-PAGE. SDS-PAGE will tell you how much protein you have. If you have additional bands, then the protein is contaminated with other proteins. If you don't and size matches, that means the protein is being purified. So let's revisit some of the important steps. For example, what we have discussed in this case was the protein purification, bacterial protein purification, or recombinant protein purification, whatever you want to tell it. It's a simple project, and most of the biotechnology students, whether they are in undergraduate classes or postgraduate classes, they should definitely perform this particular project and understand the basics of protein purification. In any basic molecular biology lab, you can do this, and you don't need fancy stuff. Yes, you definitely need some of the equipments, and they are routine, but you don't need too expensive things to complete this project. You need a plasmid, you need a bacterial cell, you need the media plates, you need to know how to lyse the cell, you need to know how to grow the cells. So basic equipments you need. And what you need to do is, first, you have to purify the plasmid. If you have a pure plasmid, transformation, it will take at least 1 day. Transformation will take 1 day. After if the transformation is successful, you'll get the colonies. And after getting the colonies, you can simply do colony PCR to find out whether the colonies, they have the specific colonies, they have the gene of interest in it. Or you can directly do the protein expression using SDS-PAGE. Nothing is stopping you, but it will definitely take time. If you want to screen out which colony is producing the protein, you can definitely, do the small growth of the bacteria in maybe 10 tubes. After that, you can simply run the SDS-PAGE gel, and you will see proteins will be there. And once proteins are there, then you can do the batch growth, and it can go up to 1,000 liter, right? Nobody is going to stop you in making that the huge batch growth, but you need the facility. In the lab, usually we don't have the facility for 1,000 liters. You can start with 100 ml or 500 ml, and then using that, you can do the column chromatography. And I think, with my experience, you'll get the significant amount of protein that you can use for your further experimentation. And what are those experiments? You can do a lot of things with proteins. If you're purifying enzyme, you can do substrate activity. If you're purifying some protein that you want to understand the DNA protein interaction, you can do further hybridization experiment, IMSA experiments. If you want to find out whether the protein, what is the sequence of the protein, you can do multitop. So there are so many things, but definitely you need a purified protein to do all those things. So this was a brief, I would say, stepwise introduction of the bacterial protein purification and what are the things that you need to do. This is one of the most fundamental technique, and I think this will stay. You can't completely automate this process, so it is worth to understand how you purify the protein. Right now, the process that is getting automated is the cloning. Cloning nowadays is getting easier and easier, and you can just get the plasmid with your gene of interest. You have to just give the sequence and you'll get the plasmid. So there are companies available that can synthesize your gene, and then you'll get the gene inside the plasmid vector, and they'll give the whole construct to you, maybe in a week. There are also some companies that are purifying the proteins for you, but it's a huge project. So I'm saying that the cost will be increased, and if you know how to purify your protein, you will be asset to those companies. So I think this is important. If you understand how to purify proteins, even with the bacterial cell, you should know how to produce or express and purify the protein in yeast, mammalian, insect cells, and that's the core molecular biology. So I hope that this video was helpful for you to understand, and at least especially the bacterial protein purification and expression part. If you want to watch other videos that I made on, for example, this blue-white screening, you can also watch that. I made video on his tag, MIC tags, and peptide tags. You will find all those videos in the cell biology series. Explore more. There are tons of videos on the cell biology topic, microbiology topic, and I hope that those videos, they're gonna at least give you the kickstart on important research topics like this one. So we'll meet in the next video, where I'll try to explain some other topic that will help you in your exam, in your projects. And if you like the content of this channel, then please, I request you to subscribe the channel and also share these videos with your friends. Thank you so much.